Hello, my name is Truls Hansson. I'm a PhD student in the MANTEL project, where I'm mainly situated in Germany at the Leibniz Institute of Freshwater Ecology and Inland Fisheries, as well as in Ireland at the Dundalk Institute of Technology, and I've been also working at the Marine Institute of Ireland. Here I'm going to talk about one of the studies of my PhD called the bright side of brownification and the dark side of light. Methane is a potent greenhouse gas with up to 28 times the warming potential of its more famous counterpart CO2, which makes it a pretty vital component for understanding and predicting the effects of global warming. It is released into the atmosphere by a variety of different sources, both natural and man-made. However, a significant portion, about 20%, is estimated to originate from lakes and rivers. Surface-to-air fluxes of methane depend greatly on surface methane concentrations, hereafter SMC. Understanding the drivers of SMC has therefore become a key point of interest. While traditionally methane production was believed to be a purely anoxic process, extensive evidence has recently demonstrated that the production of methane in near-surface oxic condition is possible and in fact is quite common. While there is still some debate regarding the underlying pathways of this phenomenon, a major part has been shown to be related to primary production. Additionally, recent studies have shown that oxidation of CH4 methane through methanotrophy mainly can be influenced by light through the process of light inhibition. Thus, light seems to be a pretty vital component driving surface methane concentrations and subsequently surface-to-air methane fluxes. However, in spite of these processes indicating that light should be a vital component, the overall relationship between surface methane concentrations and light climate, including processes such as brownification and the far-reaching urban lighting known as Skyglow, has received relatively little attention. So, first, a bit of background information on the main processes we've been looking at, starting with brownification. Brownification is most commonly defined as an increase in color of lake and stream waters brought on by color-dissolved humic matters of both terrestrial and wetland origin. These humic matters mainly absorb short wavelength solar radiation in the visible spectrum, leading to potentially strong effects on primary production and other light-sensitive processes. One aspect of brownification, which makes it highly relevant from a MANTEL viewpoint, is that it is a common result of several types of extreme weather events, with precipitation, droughts, and others all having the potential of inducing brownification through different processes. In fact, the IPCC has predicted that brownification of lakes and rivers is likely to increase in the future via climate change. The second phenomenon of focus in our study is skyglow. Skyglow is typically defined as weak illumination of the night sky and can be induced by several natural sources, but is also often mentioned as a vital side effect of light pollution. In the latter setting, urban lighting is refracted in the atmosphere and can thereby influence much greater areas than those immediately affected by artificial illumination. Because of increased urbanization as well as cheaper LED lights making lighting more affordable, skyglow is having an increasing influence on previously untouched parts of the ecosystem. The hypothesis of our study is related to the overall drivers of surface methane concentrations, namely brownification and skyglow. As previously discussed, multiple studies have demonstrated a relationship between oxic methane accumulation and light-sensitive processes. Both oxic methane production through primary production and light-limited methanotrophy should in theory limit surface methane concentration under limited light conditions as compared to control groups of less light-limited climate. We therefore hypothesized that brownification would lead to a reduction in surface methane concentration. Secondly, and based on the same base principles, we hypothesized that the addition of Skyglow would mitigate some of the effects of brownification with regards to surface methane concentrations. Finally, we expected to see light regime influencing the relationship between surface methane concentrations and one of the main culprits of oxic methane production, namely cyanobacteria. Because of increased nutrient availability induced by decreased primary production, one would expect to see a positive relationship between cyanobacteria and surface methane concentrations in treatments with more light than in light-limited treatments, i.e. a more positive relationship in the brownification treatment with Skyglow than in the one without Skyglow. In order to investigate these hypotheses, we conducted an 8-week sampling of surface methane concentrations in conjunction with the ILES Mesocosm experiment of 2018. This experiment consisted of 4 treatment groups with 5 mesocosm enclosures per treatment. Skyglow was simulated using LED lights with an output similar to that of Skyglow, while brownification was simulated by mixing human feed into the enclosures. Sampling was conducted once per week throughout the experiment, and samples were processed and analyzed in the nearby laboratory.
Data analysis was carried out using general additive mixed models, an iteration of general additive models using the MGCV package in R. The reason for using general additive mixed models is that these provide a good balance between flexibility and interpretability. It also enables dealing with problems such as autocorrelations in the serial residuals, a common issue when dealing with time series. Further, it enables users to account for other structures in the data, and thus is ideal for finding hidden patterns that other models miss. Below is an example of serial ACF residual plots with autocorrelation evident in the serial residuals of the left figure, while in the right figure the problem has been dealt with effectively by adding a correlation structure to the model. For these reasons, GAMs are increasingly becoming the tool of choice for many mesocosm studies. While being indistinguishable from the other groups at the T0, surface methane concentrations in the brownification treatment became significantly lower than all other groups as soon as the treatment was added, as can be seen in the temporal trends of each group in the figure to the left, and in the partial effects on surface methane concentration by each treatment in the figure to the right. Interestingly, surface methane concentration in the brown plus Skyglow group did not change after the treatment, namely brownification in combination with simulated Skyglow, was initiated. However, it did differ significantly from the other treatments with regards to its temporal development. Unlike the other treatments, brown plus Skyglow maintained a relatively high surface methane concentration for several weeks into the experiment, but then dropped to levels of concentration more similar to the other treatment groups at around week 5. Additionally, our observations suggest an effect of moonlight on surface methane concentrations, as enclosures in the control groups and in the brownification treatment experienced a temporary increase in surface methane concentrations during days shortly before and after the day of full moon. That moonlight might influence lake ecosystems, likely through changes to microbial dynamics, are supported by findings from previous ILES experiments by Jeremy Fonneville et al. Paper in PrEP, who found that moonlight, in addition to Skyglow, influenced the microbial community dynamics of enclosures. One of the advantages of cooperating in large-scale experiments, such as the ILES Lake Lab experiment, is that one has access to a wealth of environmental variables. This provides ample opportunity to study potential underlying patterns and to address later questions which may arise as data is processed later on. However, it also means that when attempting to explain observations, one must be extremely careful in picking which variables to include in any study. Limiting number of variables by excluding ones that are correlated is a first step in any quality control before analysis. However, even when excluding correlated variables, a good deal of background knowledge on any relevant subject is required in order to pick out which of the remaining ones are most relevant to include in further model selection. To better understand what drives surface methane concentrations, we conducted an AIC-based model selection using a pre-selected set of variables to find an optimal model. Through this process, we found that cyanobacterial biomass, pH, bacterial protein production, and nitrate together explain the largest amount of variation in our data. Because physical variables such as wind and temperature are well known to drive much of the variation in surface methane concentrations, these were compensated for but not included as influential variables in our model. It should also be mentioned that while nitrate did provide a slightly better IIC score than other nutrient-related variables, it was largely interchangeable with soluble reactive phosphorus. Of the driving variables identified through model selection, the nonlinear relationship with cyanobacterial biomass appears to be the most persistent and explain the largest amount of variation by itself. When looking at this relationship by individual treatment groups, however, we find that the brownification treatment differs from the rest with a significantly negative relationship. Interestingly, cyanobacteria in the Skyglow treatment had no significant correlation with surface methane concentrations by themselves. However, when an interaction with nitrate, SRP, or light was included, a significant positive relationship was found. Another interesting finding was that cyanobacterial biomass in the brownification treatment did differ significantly from the other treatment groups, and indeed was the only one of the driving variables to do so. In fact, of all the variables we looked at, cyanobacteria was the only one to reflect the pattern of surface methane concentration across the treatments, which might indicate that varying oxic production under different light regimes could be the only underlying driver of our main observation. This would reflect findings by previous studies, such as those made by Ionescu et al. From 2018 and 2020. Unfortunately, as we were unable to measure rates of oxic production or oxidation due to the workload during the experiment, this hypothesis remains speculative. In conclusion, this study confirms the hypothesis that brownification is capable of significantly reducing surface methane concentrations, a finding that by itself, due to its high relevance for aquatic methane emissions and the prediction of increased rates of brownification in future lakes, provides some sorely needed good news in the story of anthropogenic warming. Unfortunately, our second main finding somewhat dims this flash of optimism, as it demonstrates that even very weak levels of light pollution, a rapidly increasing global issue, is sufficient to mitigate most of the aforementioned effect. Our third finding suggests the existence of a potential turning point, possibly related to day-length induced light availability, at which Skyglow, at least in very low levels, is no longer sufficient to mitigate surface methane accumulation. Finally, our study finds supporting evidence for the relationship between surface methane concentrations and cyanobacteria. Furthermore, while previous studies have looked at the relationship between light oxic methane accumulation and cyanobacteria along with other primary producers, ours is the first to find evidence of a nonlinear relationship driven by different relationships under different light regimes in a large-scale outdoor setting. Furthermore, while we can't say anything definitive on the underlying mechanisms of the light-driven differences between the treatment groups, significant differences between cyanobacterial biomass reflecting the surface methane concentration differences between treatment groups does serve to inform future studies as well as current literature on the topic of oxic methane paradox. Finally, I would like to sum up additional project in my PhD, both published and as to yet unpublished work. My degree has been focused on the effects of brownification and extreme weather events on trophic dynamics and carbon cycling with a focus on methane in various lakes and catchment systems. The first publication of my PhD, Environmental Drivers of Mixotrophs in Boreal Lakes, focuses on the drivers of phytoplankton trophic strategies, with a main focus on color-dissolved organic carbon, as well as CO2 characteristics of mixotrophy-dominated lakes, and the influences of preferential grazing of zooplankton. The third study of my PhD includes a look at the peatland-lake interactions with regards to methane dynamics and emissions, where we investigated the potential of peatland-fueled lakes as an alternative pathway for methane to escape from the deeper confines of peatland ecosystems. Finally, I would like to thank the co-authors of this study, which includes my supervisors, Professor Hans-Petter Grossart and Professor Eleanor Jennings, as well as the participants in the ILES 2018 experiment. Additionally, I would like to thank all the people who helped out during the experiment and also the previous and latter work of my PhD.